acids. Chemistry is still important for a lot of this because even with carbohydrates, we talked about, the condensation reactions and that we can have or like, for example, the esterification, or in that case, it was the hemiacetals that we formed with the carbohydrates. The chemistry is still important there, but once you get into amino acids, there's still important chemistry that tells us how these things are formed. But I think this kind of starts to fall into the role of biochemistry because now we're linking how does the chemical properties affect like biological function, and how does structure get affected by the chemical properties, and the different reactions that build those structures affect that. Okay, so chapter 17 in the book, if you're still following along, goes through amino acids, proteins, and enzymes. And today we're probably going to try and get through most of amino acids. If we can, we'll talk about protein and enzymes more when we get back. And so, where do we see proteins? Well, proteins make up about 50% of your body weight by mass. So, compared to water, it's still a small fraction. And because they tend to have heavier elements like carbon, nitrogen, and oxygen, that means that the relative number of proteins is small compared to water molecules. But it's a significant portion of your body and pretty much involved in every process within your body, right? So, for example, your eye has different proteins that make up the gel that make up your eye. And if you have something where those start to crystallize, we talked a little bit about crystallization, then that'll cause things like cataracts and stuff in your eyes. And so whenever I've mentioned proteins and stuff before, I usually say proteins, the function is determined by the structure of proteins. So like how important is the structure of proteins? So here we have two proteins. They're not specific proteins for any disease, but it's an example. And so this would be the correct way that a protein is supposed to fold, right? So proteins, polysaccharides, they can form the linear or branch structures, right? So they're relatively simple in the number of ways that they can fold. When you're looking at proteins, they can start to form very complicated structures, and there's a hierarchy of the different structures that we can form with proteins. So this would be what the correct three-dimensional shape is for a structure. If we're looking at this, usually when you see the ribbons like this in a spiral, we'll talk about that in a little bit. That'll be a secondary feature called alpha helixes. And then when you see things kind of going back and forth like this red part over here, those are called beta sheets. And these are both forms of protein, and we'll talk about this more when we get back to spring break, called beta sheets. And so as these proteins are being synthesized, right, there's different intermolecular forces that are controlling how they interact with each other as they're being made, and that causes the proteins to then fold on themselves. And they fold into a configuration that makes everything happy, and the native state of the protein gives a specific three-dimensional shape for the protein, and then that determines the function of the protein. If you have, for example, a misfolding in that protein, you can say, okay, whatever, the body will just digest it, we'll get rid of that protein, we don't have to worry about it anymore. However, sometimes a protein misfolds in a very specific way, like in the example here. So it's like a long strand of the protein. We had these long stringy strands here and a little alpha helix over here. They misfolded and they formed a relatively kind of rigid structure here in this. It's called a beta helix, where it's like a helix of beta sheets. And so this misfolding can cause diseases, because if this one protein misfolds, and let's say it touches another protein of the same type, it can cause that other one to misfold. And so what happens is, if we were to look at, we've seen these energy diagrams where we have products and reactants. If we have our unfolded protein up here, and then we go to our correct protein, or then we have our incorrect protein, so our misfolded protein, they're very close in their energy level. It just takes a little bit of energy, which is what's represented right here, the activation energy, to go between these two structures. However, once it gets misfolded into the other structure, it can be very hard to get it to go back. And this can induce the misfolding of other proteins. And so if you guys have heard of mad cow disease, for example, that comes from eating beef, that mad cow disease is caused by a misfolded protein. where basically you eat something and just getting it in your digestive system, then eventually into your bloodstream. If it comes in contact with certain proteins in your brain, it causes mad cow disease, right? So these are, and then, I don't know if you guys have heard of it, it's kind of a big deal right now. It's called like, what's the common name for it? Zombie deer disease, where you have these zombie deers going around. That's also a prion disease, and a prion is just a misfolded protein. And it's very contagious, even though it's just a protein. It's not like it's a virus or a bacteria, which is something that's trying to replicate itself. It's contagious because if there's a dead deer and another deer comes in contact with it, and the prion proteins that are misfolded in that deer, they can cause the same misfolded proteins in the deer. So that's why, sometimes eating like brains and stuff like that is bad, because that can cause misfolded proteins in your own body. So it's very important to understand the structure of the proteins, and then the structure then determines the function. You have a misfolding of the structure, it can have catastrophic consequences on a larger biological scale. So that's why, we study proteins. Proteins are very important. Okay. And so we have a hierarchy of proteins, and this is the phrase that I've said over and over. The function of a protein is determined by its structure. And so at the same time, when you guys were learning about chemistry for the first time, generally you start talking about atoms and electronic structure in atoms. Then you take a step back, like you broaden the microscope, and you look at then how atoms interact with other atoms, and then you take a step farther back, and kind of where we're at is how do molecules interact with other molecules. With proteins, it's kind of this, a similar idea to breaking down the structure. You have the sequence of specific amino acids, which build proteins, and then that goes back into a secondary type structure, like an alpha helix or something, and then those form a larger three-dimensional structure, like the ones we showed on the previous page, and then those three-dimensional structures can interact with other three-dimensional structures. So there's a hierarchy to the protein. The smallest building block for a protein are amino acids. Right, so amino acids is what we'll be talking about today, are the building blocks of proteins. And so they can be just as complicated and numerous as the different saccharides we talked about with carbohydrates. The specific chemical nature of these will be a lot different though. And because of the structure of individual amino acids, you can get a lot more variation in the structure of proteins compared to polysaccharides. Where polysaccharides are more branched or linear, proteins can have specific shapes in three-dimensional space. Bless you. And so the structure hierarchy is we start off with primary structure, then we go to secondary structure, then we go to tertiary structure, and then we go to, does anyone know what the last one would be? Quaternary structure. I'm pleasantly surprised you guys were able to get that one, and maybe you've heard it before, but we talk about all these prefixes, but, tetra, so sometimes you would think it'd be one of those. But then it's quaternary structure. And so the same way when we add amines, primary amine is only bonded to one R group, so in a sense it's the simplest kind of amine. And secondary, we add one. With protein structure, we start with primary, then it gets larger, secondary, tertiary, and then quaternary structure. Everything all right? Okay. Okay, I was just curious what that. I was like, oh, that's an interesting alarm. All right. And so, what are the definitions of each of these? We have the specific sequence, of amino acids. Right, so that's your question?
Sorry.
No, why do I don't ever apologize for asking questions. I love questions.
Is it an amide that it's going to be attached
it's going to be an amide bond that attaches amino acids to other amino acids, right? So primary structure is held together by amide bonds, covalently linked molecules. So that's a good thing. I'll add that here. So this is a specific sequence of amino acids. These are held together by amide bonds. And sometimes a specific amide bond you'll find is called the peptide bond, right? So I know with carbohydrates it gets confusing because we talk about carbohydrates. Ketoses, aldoses, then we talk about saccharides, and then trisaccharides, polysaccharides. Proteins are amino acids, and then each of those can be classified as a single peptide is another name for it. Polypeptide is another name for a protein, right? So a lot of different ways we kind of say the same thing. All right, so that's a specific sequence of amino acids. This is like when you have a DNA sequence, and you have A, U, 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 A, T. I'm forgetting a letter, but you get the idea, right? That tells
ACUGT.
ACUGT, yeah. Oh yeah, so I did RNA because I added the U in there, right? So I did. So it's an RNA sequence, right? You have U in there. Primary sequence is like that, except instead of the bases that make up DNA, it's the individual amino acids that we have, the naturally occurring amino acids. The secondary structure is going to be structures formed from intermolecular forces within a polypeptide or protein. Right, so these are intermolecular forces. So a lot of times we think about intermolecular forces that happen between two different molecules. Well, when molecules get really long, they can have intermolecular forces that then occur within a single long chain. And so examples of these would be an alpha helix, right? So that's the Greek letter alpha, and a beta sheet. And so we'll talk more about the specifics of these, like I said, when we get back. But these are basically the primary forces that are going to be holding these together. A lot of times are hydrogen bonds. Right, so these are held together by hydrogen bonds. Alpha helixes are held together by hydrogen bonds along the protein backbone. Right? If we have an amide, what is an amide made of? What are the two things in an amide? There's a nitrogen. There's an oxygen there too, right? So there's nitrogen and oxygen. It's basically an amine and a carboxylic acid that come together, right? So that oxygen and that nitrogen has a hydrogen to it, so that can participate in hydrogen bonding, and that can coordinate with the oxygen or another amine molecule. So alpha helix and beta sheets are held together mostly by hydrogen bonds. Mostly by H bonds is how these are held together. And then tertiary structure is the three-dimensional shape of a single polypeptide chain. Right, so now we're looking at something like what we looked at when you looked at the misfolding of that protein. That structure you saw was a single chain, but it had some alpha helixes in there. Then it came around, had another alpha helix, and the misfolded one then had beta sheets that then formed from that point. So this is a three-dimensional shape now. All of those things make together, right? So they then interact through more generic. They can interact through hydrogen bonding and stuff, but more generic things like polar versus non-polar interactions, right? So the polar stuff will kind of stay together, the non-polar stuff will stay together, but they'll keep each other separate. And so these are also determined by intermolecular forces and mostly pole/nonpolar, and nonpolar forces is another way of saying van der Waals forces. And a lot of times in the context of proteins, you hear people say hydrophobic versus hydrophilic type interactions. Polar, nonpolar, IMFs, and so again, the tertiary structure is then the three-dimensional shape that the protein makes. So if you're making a protein, this is kind of like the full walking path as each amino acid is added to the chain, and you're just kind of walking through three-dimensional space. And then quaternary structure. Quaternary structure is then multiple polypeptide chains that are interacting with each other. With each other. And this isn't something that's required for every protein, right? This is going to be larger proteins where you have multiple parts of multiple different chains interacting with each other. But it isn't true for every single protein. A lot of times you can have smaller proteins. So for example, a lot of virus shells, the simplest viruses, the little proteins that make up the shell that the viruses surround their RNA or DNA with, that is usually a protein structure made up of a single chain because it's a small little virus. But when you get to larger viruses, like HIV, for example, you can have protein shells that are very complicated, and you have one fraction of the capsid protein or the shell protein that then has 4 different polypeptide chains that all interact with each other to give it a unique shape. But then, for example, in the simple viruses, whatever interacts with the genetic information, whether it's RNA or DNA, that can be a single polypeptide chain. All right, so these are the different classifications for the hierarchy of structure that we have in proteins. And so we're going to kind of start from primary structure, especially once we get into polypeptides. Today we're probably not even looking at primary structure because we're just going to be talking about amino acids specifically, which is the building block of proteins. Okay, any questions on the structure? No? All right. Okay, so again, amino acids, these are the building blocks of proteins, the simplest unit for proteins. And these can also be called peptides. This is another name for an amino acid. And so, like what Sarah was asking before, generally amino acids are snapped together by amide bonds, right? And so each amino acid, for it to be combined in a condensation reaction like that, needs to have one side that has the amine, and the other side has to have the carboxylic acid, right? It's like an extension cord. You have one part that plugs into the wall, and one part that plugs into whatever you need to be plugged into the wall. So the amino acids, we have the N, actually let me do green for that, the N and the C terminus, right? The N terminus is going to be where we have the amine, part of the amide bond, and the C terminus is where we have the carboxylic acid side of the amide bond. And so if we were to draw this out, if we have an example, so this is a generic amino acid, well, something like H3N C double bond O, and then here O minus, and here we have R. So this is the kind of basic, most fundamental structure we can have for an amino acid. Where the nitrogen is, there's going to be a positive charge here. This is going to be the N terminus, or the N terminal, and this is a negative charge over here. This is the C terminus. And the reason it's called the C terminus instead of the O terminus, you might be wondering, is because, well, the carbonyl carbon is actually what snaps to the next protein, right? We have an amide, which means that we have to have an N that's directly bonded to a carbonyl carbon. As you can remember, when we do these, for example, in the esterification, we had an alcohol and a carboxylic acid, so we have to lose an oxygen somewhere. In this case, we're losing the oxygen that's coming from the carbonyl carbon. In the carbon that's attached to the R group, so here we have the R group is called the alpha carbon. And so here we have 4 different groups that are bonded. Sorry, there's an H there as well. 4 different groups that are bonded to this carbon. So this is going to be a chiral center. So, you know, skipping ahead a little bit, you can imagine there's going to be some stereoisomers here. It's called the alpha carbon, and the R group is responsible for the identity of the amino acid. So this part of the protein forms the protein backbone, right? So everything I've drawn here. But the R group is what determines the identity of the amino acid or the peptide. So everything that I highlight here right? That's the same for any amino acid. Well, mostly the same. There's some amino acids that the R group will coordinate with that amine. And so because these are single bonds here, that means that each of these groups can rotate around these single bonds, and that's what allows the protein to adopt a lot of different 3-dimensional configurations here. Do you have a question, Sarah? You look like you have a question, don't you?
Yeah, well, I guess I was a little bit confused, or maybe it doesn't matter, about the carboxylate ion or late ion
Yeah.
versus acid.
Yes, that's actually very, very important for protein structure. And so that's towards the end of this lecture. We talk about how do we determine whether it's protonated or deprotonated, right? If we have a carboxylate ion or the, ammonium, the positive charge there, how do we determine that? So we do determine that, and that's very important because if you have a charge there, that means it has different interactions with other molecules, and that affects very much how the shape is formed, right? So that's why in your body, your cells keep a consistent pH, because the pH affects whether or not an acid is protonated or deprotonated. So your body keeps a very consistent pH, because if you lower the pH too much, certain amino acids are going to start to deprotonate, and it destroys the structure of your proteins. So yeah, that is something important to keep in mind. Right now, I just have everything charged, but the total charge on this is neutral, so we'd call it, this is at its isoelectric point, or the Zwitter ion, which is, we'll talk about that more. Okay. So amino acids also only have common names that are based on their R group, right? So I know, which, is both a good thing and a bad thing. It means it's kind of, you don't know the names, it's kind of hard to determine what the structure of the protein or the amino acid is. You don't have the names memorized. But it's a good thing because I'm not a big fan of memorization of the different names, so it's not something that I'm really going to be emphasizing that you guys do. It will be easier if you do memorize the names. And so that name, again, is based on their on their R group. And so the name is kind of broken up into 3 different ways that we can name them. We can have the full name, a 3-letter sequence that tells us which it is, and then we also have a single letter representation. So that's kind of similar to DNA bases, but then we also have a 3-letter sequence. And so if we look at, for example, glycine, the 3-letter sequence would be GLY, and then the 1-letter sequence is going to be G for glycine. And so you can see there's going to be some weird ones in this case because there's a lot more amino acids than there are bases in RNA or DNA. So this, and a lot of them start with the same letter. So then you have to come up with different letters, right? So there is a, alanine has a 1-letter sequence of A, but then there's also arginine, right? So they both start with A. So arginine is represented by the letter R. That one's a little easier to remember. That's why I remember it, because arginine, R, pirate. Right, so there's really no way around these ones. These ones you kind of just memorize the 3-letter sequence, the 1-letter representation, and then the full name. So this would be a good, idea for this type of thing to do flashcards for. Okay, and so we'll go through half of this slide and then we'll come back. We'll take like a 5-minute break. We'll come back and keep talking. So amino acids are usually classified based off of their physical properties. Right, so when we think about physical properties of molecular compounds, what types of things are we usually thinking about? So yeah, geometry, structure, how it interacts with other things, and how it interacts with other things is determined by what topic keeps always coming up. Intermolecular forces, right? So the intermolecular forces is largely how we classify these different things. And so because they all have that same backbone of the N-terminus, those 2 carbons, and then the C-terminus, right? What do you guys think the physical properties are mostly determined by? It should be the R group, the same way the name was determined by it, right? So if we have a different R group, then we have different physical properties. And we'll go through some examples of doing this. All right, so we'll go through the different classifications. Let's take like a 5-minute break, so we'll be back at like 11:27, and then we'll keep on talking about amino acids. Go ahead and continue. All right, so the different classifications are going to be non-polar side chains. There's going to be polar uncharged side chains, or R groups. And there's going to be polar charge groups. And so the nonpolar, I'm not worried about splitting them up, but sometimes you'll see it as aliphatic, nonpolar, which just means it's going to be like a alkyl chain. And there's also aromatic nonpolar. And they make that distinction just because aromatic compounds tend to have unique properties, and it's important for biological function. In general, I'm not going to ask you to identify between those two. It'll be more, is it polar or nonpolar? Polar uncharged is pretty straightforward. It's just going to be if the side group is polar, right? So that means we have a permanent dipole in that side group. And then uncharged, meaning that it's not going to have a positive or a negative charge on it. Polar charged can either be negatively charged or a positively charged R group. All right, so these are the different classifications. And again, so they're classified based off their physical properties. Their physical properties usually puts them in specific spots. So for example, some different R groups, which then gives the identity of the amino acid, are going to be in specific motifs within proteins, right? So you probably see a lot of alanines. For example, an alanine is going to be just a methyl group as the R group. It's a relatively simple group, not too big, not too bulky. You see that a lot in alpha helixes because it doesn't get in the way and allows it to form a nice helix. The structure can form a nice helix nicely. Whereas other ones, if you have big bulky side groups, those might be in certain motifs, like a turn in a chain or something like that. Maybe they don't have a specific structural motif. Okay, so that's how we classify these. All right, so I'll give you guys a second to think about this. So here we have 3 different amino acids. Let's do the first one together. We have threonine, right? So if I highlight the part that is the kind of backbone of what would form a polypeptide, so that's the same for every single amino acid for the most part. What's highlighted in red, and what I'm highlighting in red right here, this is the R group, and that's what makes this amino acid threonine. I'm gonna use a different color because that's kind of hard to see. Maybe blue. That's red. Right? I already used blue. That's going to be the R group right there. And so here we have 2 carbons that are bonded to each other and then an OH group on the second carbon. That OH is probably going to make that a polar bond right there, right? So we say that this would most likely be a dipole moment there. Moment from the carbon-oxygen bond, and that means that this would be a polar R group. And one thing I'll specify for the uncharged versus the charged polar groups is that's usually referencing physiological pH, right? So that's at whatever your body's pH is naturally at. You can always change the protonation state just by changing how acidic or basic the environment is. All right, so that's going to be a polar R group because we get a permanent dipole moment from the carbon-oxygen bond that's in there. At physiological pH, and we'll talk about that in a second, it's not charged. All right, so why don't you guys do the same thing then for arginine and leucine here. Determine if they're polar uncharged, or non-polar. Arginine first, right? So arginine, how do you guys classify it? Polar charge, right? So generally, if there's a charge there, there's maybe 1 or 2 counterexamples of that. But if there is a charge, then it's polar. So generally, if there's a charge, it's going to be polar, right? Because that means on one atom, there's a whole difference in an electron not being on that atom, which means that other atoms have had to take that, so that atom is electron deficient if it's a positive charge, and the other atoms are going to be electron rich. So there's probably going to be a dipole that goes something in this direction, right? So this means that it is a polar charged amino acid because there's a positive charge right there. There's a positive charge. That nitrogen has 4 bonds to it. We can calculate the formal charge. If we remember, the number of valence electrons, which is 5 for nitrogen, minus the the number of bonds minus the number of lone pairs. In that case, it would be 5 minus 4, so you just get a charge of +1. Okay, so that one's going to be polar charged. So arginine is one that I looked at a lot over the years. So this is one that a lot of viruses have on the tails of their capsid protein. Because there's positive charge, DNA is negatively charged. It's used to kind of condense the DNA inside the capsid. All right, what about leucine? How are you guys going to classify this? So leucine, if we were to name that group, that R group, what would you guys call that group on the leucine? How many carbons are there?
4.
4. And so what's the prefix for 4? Bute, right? And then this specific isomer is isobutyl, right? So this is like an isobutyl group that's attached to the amino acid, right? If it was propyl, then there would be 1 less carbon in the chain that reaches the, the peace sign at the end. So this is an isobutyl group. And when we see alkanes, right, if we see something like this on an alkyne chain or something like that, do we usually say that that's a polar? No, it's just an alkane chain, it's just carbons bonded to carbons. There's no electronegativity difference, just hydrogen. So this is going to be an alkane or alkyl chain. And alkyl chains are nonpolar. Right, so you'll see something like a leucine on the interior of a lot of proteins because it doesn't want to be in the water that's surrounding the proteins in the cell. So a lot of leucines will be on the interior because it has more hydrophobic type interactions, which really just means that it has only van der Waals type forces. So this is nonpolar. Polar charge. And this one was just polar R group, and then also the first one was non-charged. Okay, any questions on that? Right, so this, if you want to brush up on intermolecular forces, this is how we can determine if the amino acid is polar, non-polar, or polar charge, right? And so these, if I was to ask questions about this type of thing, it would be, okay, where would you expect this to be on a protein if it's floating around in the cell, on the interior or exterior, based on how it might interact with water, for example. Okay. So, as with everything else we've talked about up to this point, there's stereochemistry, or we have stereoisomers. These are going to be the same as for saccharides or carbohydrates. That means that we have both L and D isomers, right? And what type of specific stereoisomer was that? Right, we have structural and then stereoisomers. Structural is different connectivity, but the same atoms. Stereoisomers was the same connectivity, same atoms, and yeah, that's exactly right. So it's going to be an enantiomer, but different 3D spatial arrangement, right? And so these were called enantiomers. Same connectivity, same atoms, right? So the isomers, they have to have the same number of atoms and the same of each type. They're connected in the same way, meaning you still have the same number of carbon. Each carbon still has the exact same connections. It's just the way that they're arranged in three-dimensional space is different from each other. All right, and enantiomers, specifically were chiral stereoisomers, meaning that they have a chiral center in there and they're mirror images of each other. And so the only difference is with amino acids is in carbohydrates, it was the penultimate carbon that was what determined if it was D or L, and it was the orientation of what group? Yeah, it was the OH group, right? The alcohol on the fifth carbon, for example, on glucose or fructose is on the fifth carbon down. It was the orientation, whether the OH group is on the left or on the right, right? The only difference for amino acids is it's going to be the orientation of the amino group on the alpha carbon instead of the penultimate carbon. And opposite to sugars, most natural amino acids are in the L configuration, right? There's a couple that occur in some bacteria and whatnot that are in the D configuration, but most naturally occurring amino acids, right? And that's just what natural means, right? So a lot of, modern medicine and stuff like that, we can synthesize non-naturally occurring amino acids. You can put whatever R groups you want onto an amide. They're going to be in the L configuration. And so how this would look is it would be almost identical to the Fisher projection for a polysaccharide or a saccharide, except instead of the aldehyde or the ketone at the top, we have a carboxylic acid, because amino acids have the C terminus, which is where the carboxylic acid terminus is. Then we only have one group here, which is going to be the amino group. And I'm gonna keep this just as negative because we haven't talked about them being positive or negative there. And then what's at the bottom is just going to be the R group. So in this case, it's going to be CH2, 3, and HC. That's what arginine looks like, for example. So really the only important thing, bless you, right? This is as big as your amino acid Fisher projection is here. The R group is relatively unimportant in terms of determining whether it's a stereoisomer or not. You may as well just leave that as R. All right, so when we compare that to our saccharide, which was the polyhydroxyl, at the bottom we always had the CH2OH. And then we could have any number of carbons in between. And then everything else was OH, CH2OH. Right, so for the saccharides, we either had aldehydes or ketones, and that was it. For amino acids, it's a carboxylic acid or carboxylate ion that's at the top. The bottom is only going to be one carbon away, right? This is going to be our, what was the name of this carbon again? The penultimate carbon was, these ones are called the penultimate carbon, right? This was the penultimate carbon. In both of these cases was the penultimate carbon. In the amino acids, that was called the alpha carbon. The alpha carbon is the one that has the R group on it. Right, and the alpha carbon is the chiral center for the amino acids. There's only one amino acid that doesn't have a chiral center. It's actually the one we talked about on the previous page, glycine, because it doesn't have its R group is just a hydrogen. If you have two hydrogens, it can't be a chiral center. right? So at the bottom here, we just had the CH2OHs in our saccharides. Here, it's our entire R group that's down here. So nothing that's attached to the R group determines which stereoisomer that we're dealing with. It's just the orientation of the amine group. All right, any questions on that? No? Okay. All right, so let's get some practice doing that and just familiarize yourself with some of the different amino acids. So why don't you guys draw the Fisher projections for the following amino acids. Right, so you know for each of them, what are you going to have? What are you going to have for all of your amino acids at the top? Yep, we're going to have our double bonded and OH here, right? We're going to have our carboxylic acid group there. Then we're going to have our alpha carbon, and then we're going to have our R group at the bottom for each of these, right? So this is the same for each of these without considering the specific R group that's there, right? Then the L or the D configuration is just determined by what? Yep, it's by the alpha carbon and which side the amine group is. So if it's L-leucine, that means the amine group should be on which carbon? Should be on the left, right? It's the same as with saccharides. H3N, this one has a positive charge. I'm going to make these carboxylate for each of these still. Minus, minus, minus. For D, phenylalanine, it's on the right. And then for L-methionine, the left. Then the hydrogen just fills the other side.
So when it's on the right, it's NH3 plus?
It's NH3 plus on all of them.
Oh, you just wrote it.
Yeah, these ones. Oh yeah. So I've done some questions about this. So not a bad idea to point out. I usually write the chemical formula backwards if there's a bond going to it like this, because I always like to draw the bond going to the atom that it's actually
Oh,
bonded
it's
to.
like this in the end. Okay, then.
Yeah, because sometimes people you'll see something like if there's a bond from like a cyclohexane or something like this. I'll do it on the other side so it makes more sense. They might do something like CH3, and that's fine if you understand everything that's going on, but sometimes I feel that's confusing because it looks like it's bonding to a hydrogen there when it's not. It's bonding to the carbon.
Okay.
Yep. But yeah, that's good to point out actually. So then I'm going to erase each of the R groups. Phenylalanine is going to be an aromatic hydrocarbon group, an aromatic R group, and it's just having a phenyl group, which is just a benzene ring that's attached to it like that. What was the leucine group again?
A butyl.
Yep, it's a butyl and it's a specific isomer, which was an isobutyl, right? So that's what the leucine looked like. And then methionine. I'm trying to see if I remember methionine. You would think, right, because it has meth in there, but it's actually, I believe, has a sulfur in there. Yeah, so methionine has a sulfur group. I'll look it up real quick because I don't remember it off the top of my head, but it's a amino acid with a sulfur group. Methionine, I believe it's just sulfur and then a methyl group after that. Or if anyone has better internet, they can look it up for me right now. Methionine, okay. It is 2 carbons and then the sulfur. 2 carbons, sulfur, and then it has a methyl group that's attached to this. Draw that a little better. S, like that. All right, so there's 2 carbons, then the sulfur, and then we have a methyl group that's after that. All right, and just to remind everyone, when we have lines that go to atoms that are not carbon or hydrogen, this line does not mean there's a carbon right here. That means that this is the bond from this sulfur atom to that carbon atom, right? So there's 1, 2, 3 carbons total in this R group. There's no carbon there, and there's no carbon there. Those are the bonds to the non-carbon or hydrogen atom, or in general the heteroatom is what we would call that. Okay. All right, so Sarah was asking earlier about the protonation state of these different amino acids. We've seen some that have charged side groups or could possibly have charged side groups. And all that depends on is the pH of the solution that this amino acid is in. In the standard protonation state, what was the term I used, is usually referencing what pH? What do we call the pH of your body in general? You guys, some of you guys might be taking a class called. It's close to neutral, right? So it's close to neutral. Mm-hmm. Natural would be another way of phrasing it, but in general we call that the physiological pH. So I don't know if you guys are taking physiology or if you have to take physiology, but this is called the physiological pH. And this is roughly around 7.3 to 7.5 usually. And so at high pH, generally things are deprotonated. Right, as you add base to the solution to minimize the change in pH to the solution, the amino acids will start taking the proton, they'll donate their protons so you neutralize it. And then at low pH, things are usually protonated. Right? And it's not just specifically at low or high pH. It depends on what R group you're looking at, or the C or N terminus also depends on the amino acid. And so at neutral pH, and even some of these other pHs, it depends on what we call the pKa value. So you guys have probably seen that in your Chem 51 class. And the pKa value, in a simple way, the way we can think about it, is that's the value where at that pH, if you're below it, that means you're going to be protonated, and if you're above it, you're going to be deprotonated. Right, so one way, and we'll probably end with this, and I'll continue talking about this when we get back from break. But if you have your carboxylic acid, so if we have C and H3 plus OH, and then our R group is over here. This is our C terminus over here, right? The pKa for this terminus is usually around like 2.0. So that means you have to get a really low pH before this becomes protonated. right? So that's why I've been drawing it all the time as the carboxylate ion, because you have to be at a pH of 2.0, which is like concentrated hydrochloric acid, and that doesn't exist in your body, right? So you don't really see the protonated form of the carboxylic acid or the C terminus of the amino acid. At around a pH of 7, this is always deprotonated. And so it's always going to be like that. All right. And so we'll stop there. We'll continue with this after spring break. We'll continue talking about amino acids, proteins. I'll try and, I know the last couple weeks was a lot of information, so I'll try and slow down a little bit at amino acids so we can focus. And I'll try and throw in some review for carbohydrates and stuff as we're going through this. Okay, slide 10.